discussions and underscores our shared commitment to advancing in our respective fields. I look forward to productive conversations and collective learning as we embark on this educational journey together. SPSR is distinguished for its engaging webinars, further enhancing its status as a leading platform for continuous professional development in the pharmaceutical sector. I am confident that today's session will uphold our tradition of excellence, providing valuable insights from a respected speaker. Let us seize this chance to gain new knowledge, participate actively, and progress our field. Welcome, everyone. I look forward to a productive experience for all the participants. Thank you for joining us today. We are gathered today to commemorate the 102nd SPSR International Webinar on Beyond Sweetness, Unlocking the Therapeutic Potential of Honey and its Derivatives, a distinguished event hosted by the Society of Pharmaceutical Sciences and Research, SPSR, in association with Malaysian Pharmacists Society. Under the guidance of Dr. Shashi Alok, National President of SPSR, the Society continues to play a pivotal role in fostering innovation and advancement in the field of pharmaceutical sciences. The SPSR is devoted to advancing the pharmaceutical field through a variety of strategies. Our mission is to organize conferences that support pharmacy-related initiatives and endorse programs tackling public health issues in India, focusing particularly on disease prevention. We make it a priority to recognize and honor pharmacists whose notable contributions drive progress in our field. At the heart of our mission is a commitment to spreading pharmaceutical knowledge, ensuring a steady and robust exchange of information about pharmaceutical practices and services. By striving towards these objectives, we aim to elevate the quality and impact of pharmacy profession, fostering a proactive and collaborative community dedicated to excellence. It is our great privilege to introduce Professor Dr. Gan Siew Hua, Dean, Faculty of Pharmacy, University of Cyberjaya, Selangor, Malaysia, as a distinguished speaker. Professor Gan Siew Hua expertise greatly contributes to our event, providing substantial knowledge and insights. Her participation is certain to enrich our discussions and expand exploration of unlocking the therapeutic potential of honey and its derivatives. We are grateful for her engagement and anticipate an enlightening session. It is our privilege to introduce Mrs. Monika Sabharwal as the convener for today's webinar, who also serves as the founder and National Secretary of SPSR and CEO SPSR Periodicals Private Limited. Her significant involvement and leadership in the pharmaceutical sciences and research communities are indicative of her commitment to the field. We are very grateful to have her guidance in today's session. Now we will start with the short introduction to guest profile. Professor Dr. Gan Siew Hua holds a B Pharmacy from University of Manchester and both a master's in clinical pharmacy and a PhD in pharmacogenetics, pharmacokinetics from University Sains, Malaysia. She has authored over 340 Scopus Index publications and has been recognized among the world's top 2% most cited researchers for 4 consecutive years, that is from 2022 to 2025, underscoring her remarkable contribution to scientific research and writing. Her passion for academia extends across borders. She has taught at more than 15 institutions in Malaysia, Indonesia, and India, and actively serves as a trainer in research grant writing. Professor Gan has served as an evaluator for fundamental research grant scheme applications at various institutions and for the Hubbard Turin Partnership Malaysia, France, and was also invited by the Chilean government to evaluate proposals for Condicit regular grant competitions. Her research is deeply rooted in natural products, particularly honey and propolis. Through her industrial collaborations, she has received the ITEX Gold Award for Natural Product Innovation and the ITEX Silver Medal Award for a propolis formulation co-developed with industry partners. She also contributes to national standards as a member of the Honey Working Group. Department of Standards, Malaysia, ensuring the quality and authenticity of Malaysian honey. Professor Gan has authored over 10 books, including the publications with John Willey and Sons and Frontiers, and serves as an editor for 9 prestigious journals such as PLOS1, Seminars in Cancer Biology, and Frontiers in Pharmacology. Her outstanding academic leadership has been recognized through the National Outstanding Educator Award, further exemplifying her excellence in research, teaching, and mentorship. It is with great satisfaction and pride that we declare that we have exceeded 2,600 registrations for this webinar. Registrations are received from India, Africa, Algeria, Azerbaijan, Bahrain, Bangladesh, England, Ethiopia, Ghana, Indonesia, Iraq, Kazakhstan, Kenya, Saudi Arabia, Kuwait, Malaysia, Nepal, Nigeria, Oman, Philippines, Poland, Turkey, UAE, Uganda, United Kingdom, and Vietnam. We are deeply moved by the substantial and varied responses that we have garnered. Our heartfelt gratitude is extended to each person who has taken a moment to participate in this significant event. Thank you all for joining us today. Let's make this webinar an insightful and enriching experience for everyone involved. We have our esteemed keynote speaker, Professor Dr. Gan Siew Hua, with us. Welcome, madam. Your insights would be highly valuable, and I would appreciate if you could present them in a speech. Over to you, ma'am.
Okay, thank you, Dr. Pradeep, for the kind introduction and also for the society for inviting me. I will now share my slides. It's a bit noisy here, sorry. Can you see my slide?
Right, ma'am. Please make it a full screen.
Right. It's not, yeah.
Right, ma'am. Perfect view.
Okay, so I would like to start my talk on unlocking the therapeutic potential of honey and also honey derivatives. So my presentation is divided into 5 parts. The first one is on general introduction on the types of honey, because there are more than 300 types of honey available in the world, depending on the flower that the bee visit. And then the second part is on the purity of honey. How do we determine how pure the honey is? This is normally based on the international standard. And for the third part, on the majority of it will be on the medicinal properties of honey. And then I will also touch a little bit on other bee products like bee venom, propolis, and royal jelly. And finally, on Meliponiculture. I'm not sure whether you know this or not, but it's actually cultivating the hive at our house. So if you are interested. It can be in the farm as well, yeah. So the first part is on general introduction. So as I mentioned just now, depending on the flower that the bees visit, there are more than 300 types of honey. And for most studies, we will compare with standard, gold standard. And for honey, the gold standard is Manuka honey, not because it's better than our honey or Asian honey, but because it's widely researched on. So there is a lot of data on Manuka honey. So Manuka honey is normally taken as the gold standard for all honey research. So are you aware that honey is actually a combination of the flower nectar and the bee saliva? So what happens is the bee will take the nectar and then swirl it in its mouth and then swallow and then regurgitate up again and do it many, many times. I think about at least 3 to 5 times. And then it will spit out as the honey. So actually what we are taking when we take honey is the bee saliva plus the nectar. And depending on the flower, so we have many types of honey. And you can see that this one on the left side here is a stingless bee honey, which can be read at home, which is what I will be talking about later on the part 5, which is on the Meliponiculture. So do you know that 80% of honey in Asia is actually fake? It's sugar water, because a lot of people will consume honey and it's very cheap to make sugar water. So a lot of unscrupulous people take advantage of this fact. And in fact, in the Philippines, it's about 75% honey is also fake, and almost everywhere. Because honey is found on the kitchen table and everywhere, not only as a medicinal agent. Honey is the world's third most fake food because it's so easy to fake it. So that goes into our second part, where we are interested to know about the honey in Malaysia. Is it really pure? The one that is available on the supermarket shelves. So Malaysia, because I'm from Malaysia, I'm sure it's the same in India, is a country rich with flora, fauna, and has many different types of honey. And honey has been used over the years as wound healing, as antimicrobial, and also to prevent aging as antioxidant. So it has many benefits. So the first question that we had was, is the honey consumed in Malaysia from the supermarket shelf is of high quality? So we wanted to investigate on the physical, chemical, and antioxidant properties, on the content of the honey, on the pesticide, whether it's contaminated or not, and also on the medicinal properties. So what we did initially was to buy about 8 different types of honey from the supermarket shelf. And these are the types of honey that we investigated. And we compared with the gold standard, which is Manuka honey. And we will look at the pH. Honey should be acidic, the moisture content, the color intensity, the vitamin C content, the proline, or is a type of amino acid that is only available in the bee saliva. So if proline is not there, it means that the honey is fake, because no human can manufacture proline. It occurs naturally in the bee saliva. So proline is whether yes, it's available, or no, it's not. So no is not means highly likely it's a fake honey. And also the amino acid content. We also look at the antioxidant properties, the common one, which is phenolic and flavonoid, using HPLC. And this is the standard that we use from the International Honey Commission, 2002, where honey should have a pH of acidic pH of between 3.4 to 6.1, moisture content of less than 20%. If it's higher than 20%, it means that the honey is either contaminated with water, or it could be old honey. It could stimulate bacterial growth, because the bacteria like to have in a watery environment as well. So the moisture content has to be less than 20%, total sugar of more than 60, sucrose of less than 5-hydroxymethylfurfural. I'm going to talk more about this later on. This is a very interesting compound present in honey. The color intensity, the darker the color of honey, the higher is the antioxidant properties, the protein amount, and the proline, the one that I say must be present to ensure that the honey is pure. So first of all, we found that all of our honey samples were acidic, indicating that it's pure. And the pH was similar to honey taken from India, Algeria, Brazil, Spain, and Turkey. And acacia honey, among the 8 different types, was most acidic, and meaning that it has a higher stability against microbial spoilage. And Moisture content was also low in 8 of our honey types, and this can help ensure that the shelf life is within what is recommended. And Manuka honey is very viscous. It's yellow in color. It has the lowest moisture content, indicating that the shelf life can be longer because it will be resistant to microbial growth because water will stimulate microbial growth, as I mentioned just now. Then we also look at the total sugar content. It was within the recommendation of more than 60% generally. And sourwood honey had very low sugar content. And there is this belief that the diabetics patient, they should consume more sourish or bitter food to counter their disease. So it could be that sourwood honey, due to its low sugar content, may be more preferred in diabetics, though we don't investigate this fact, but we feel that if a diabetic person wants to take, probably this is one of the best type of honey. And then we found that the honey from India tend to be sweeter. We are not too sure why. It could be due to the content of the nectar in the flowers in India. I do not know. We didn't investigate on this. And the sucrose content was within recommendation. And this is the compound that I want to talk about just now, the 5-hydroxymethylfurfural or 5-HMF, which is a compound that can be present not only in honey but in any food components. Is released due to overheating, due to poor storage, or due in all honey. So the more that the honey is being heated, the more 5-HMF is being released. And this compound is interesting because it is also carcinogenic. It causes cancer. So we investigated the content of 5-HMF in Malaysian honey, and we found that, sorry about the noise, we found that if honey is stored longer than 1 year, the 5-HMF content is beyond the recommendation of the International Honey Commission. So it has increased to high levels. So our recommendation is that honey, processed honey, should be consumed within 1 year of it being processed because we do not want to have this compound in high amounts. So this is our paper on that. And then we look at the color intensity. Color means the presence of pigments such as carotenoids and flavonoids, as well as phenolic acid. So the higher color it is, the darker color honey, the higher of the antioxidant properties tend to be. So if you want to select honey, you can choose the darker colored honey. So among the Malaysian honey, again, sourwood and honey is good, and also tualang honey is also good based on our research. And these are the phenolic and flavonoid content, and this is the comparison. Again, some of the Malaysian honey has higher phenolic and flavonoid content as opposed to our standard, which is Manuka honey. Then we attempted to look at the phenolic acid present by using high performance liquid chromatography. And we found a few interesting components in rubber tree honey and also tualang honey. And in fact, our group was the first to report on phenolic acid. We found 6 phenolic acids and 5 flavonoids in Malaysian honey. So because our group was the first one to report, it won the most cited paper award in the Journal of Food Science way back in 2011. So if you want to go for most cited, you can do things that people have not found yet. And like for our case, it was we are the first to identify the phenolic and flavonoid content in Malaysian honey. Or not to really to identify, to report. Yeah. Then we look at the vitamin C content, and we found that the Borneo honey, rubber tree honey, sourwood honey, they are rich in vitamin, which will give it sour taste in the sourwood honey, for example. And for proline and protein, it's within the recommendation. And just to summarize, so we found that most of the physical chemical properties of the parameters of the investigated honey were within the limit of the recommendation. And sourwood honey and tualang honey contain the highest concentration and probably be among the best in Malaysia samples. If you want to take this type of honey, that's probably the best. And then we further on to mineral and trace element content by using atomic absorption spectroscopy. And we found that sourwood honey contain high sodium and potassium or in a balanced amount. That means it could be useful for hypertensive individuals because both sodium and potassium are high in a balanced amount. And Manuka and Gelam honey, they are rich in calcium, and this could be useful for osteoporotic individuals. And then we also found that the high sodium and potassium could potentially be due to the high levels of this mineral in the leaves itself that got transferred to the nectar because the leaves has been reported to contain high sodium and potassium as well as magnesium amount. So this is our results for magnesium, zinc, and iron. Rubber tree honey is rich in iron and zinc, and this could be useful for anemic individuals. And for zinc, it is also rich in zinc and could be useful in patients with prostate cancer or BPH and so on. Although we are recommending it to the different type of disease individuals, we didn't do the study clinical study to show that it's useful for these individuals. We are just finding that these are high in certain types of honey. So I would like to caution that we actually need to go further for the clinical trial for these types of the recommendation. We also found that pineapple honey is rich in cadmium and acacia honey in cobalt. And some of these are important trace elements that can help to, that has good anti-cancer effects. So just to summarize on the mineral content, Manuka honey is rich in zinc and calcium. Rubber tree honey is rich in iron and zinc as well, and can be useful for wound healing, acne, eczema, and also to maintain prostate health. But again, I would like to reiterate that the utilization of honey in different diseases, this need to be reconfirmed in proper clinical trials. These are our initial findings. So the next one, we investigated on the pesticide residue, and we investigated on 7 organophosphorus and 3 carbamate that are commonly used in Malaysia. And the good news is that these are not found in our 8 honey that we investigated on. Interestingly, honey is an indicator of environmental pollution because honey collected by the highway where there is a high traffic density and also by the railway track, they contain high lead concentration. So overall, the investigated 8 different Malaysia honey are pure and they are abundant source of mineral important for human diet and growth and also health. But I would like to caution not to put honey in your cooking because it will destroy all of the heat will destroy all of the good benefits, the vitamin C and so on of the honey. So you only get the sweetness of the sugar only. So next, we attempted to look at the volatile composition of honey, and the honey that we selected in the beginning was on tualang honey. And we found that methanol can pull out the most volatile active constituent from tualang honey. And using GCMS, we identified 35 volatile constituents. And a side thing that we didn't want to look for, but we found was actually, interestingly, when honey is kept in plastic bottles, the perthalate or from the plastic sizer actually leach into the honey that is consumed by human. So our recommendation is honey should be stored in glass bottles away from direct sunlight, which can stimulate the release of 5-hydroxymethylfurfural just now that I mentioned. So now I'm going to part 3, the medicinal benefits of honey. If you are a Muslim, I'm not a Muslim, the Holy Quran mentioned to drink honey from the healing jar. Yeah, that is a sign that, honey is something that warrant investigation. So our first study was, initially we wanted to study sourwood honey because I found a lot of benefit for sourwood honey. But because we found that tualang honey also has high antioxidant properties, so we targeted tualang honey. And tualang honey is a honey gathered from the tallest tree in the Asian rainforest. It's known as Cumpacia excelsa. Just a caution again, due to the modernization and the rampant chopping down of trees for development, for building and so on, we have issue with getting the source of tualang honey. And now a lot of people change to stingless bee honey, which also has high antioxidant properties. So I'm sharing with you my initial study that focuses on tualang honey first. So it is sad, our resources are dwindling and we can't really focus on tualang honey. But we found that it has good antimicrobial properties, where it has effect against, it has similar antibacterial properties with Manuka honey, but more active against certain types of bacteria. And we found that it's useful for wound infection and to boost immunity. And that's why you can see that during the COVID time, a lot of people really take honey to ward off COVID. Though again, this is not investigated, but it helps to boost the immune system. So we did in HIV positive individuals. We did this study among drug addicts, and we give honey to the drug addicts, and we found that who are HIV positive but not showing the symptom of HIV, we found that honey reduces the viral load and improve the quality of life as well as CD4 count. But of course, you may argue that the better nutrition from honey may contribute to the improvement in the quality of life. So that part we are unable to differentiate yet. So that is an interesting study to show that honey is useful for honey can boost our immune system, but not in COVID that we did. Okay, then we also look at the anti-proliferative effects, and this one we apply honey locally, and we found that it can protect against keloid fibroblasts. Because honey contains a lot of fatty acids like palmitic acid, stearic acid, oleic, linoleic acid, and so on. We also found, this is in animal, not in human, that honey protects the liver and kidney and help to reduce cholesterol. But a study in human found that honey also reduces LDL cholesterol and increases HDL cholesterol. So this is good news for people who have high cholesterol levels. But make sure that the honey is pure. Honey is also interestingly taken for weight loss. And this is because honey reduces the size of fat cells. It has lower glycemic index as compared to sugar. So it doesn't give that sudden sugar spike. Because its content of sugar is not glucose, but it's fructose, which needs to be broken down to glucose. It's a bigger molecule, and it also increases gastric emptying time. And interestingly, sugar also increases leptin levels, which suppress appetite. But don't take too much. 1 tablespoon per day is good because anything too much is no good. So 1 tablespoon a day is recommended. Can honeys help with sleep? So honey, the sugar in honey stimulate insulin release, which stimulate tryptophan release, which is converted to serotonin, which is converted to melatonin, and it is found to generally help with sleep or insomnia. Honey also protects against radiation mucositis. So those patients, for example, breast cancer patient, they can be asked to keep honey in their mouth, a tablespoon of honey in their mouth right before going for radiation therapy for breast cancer. And then actually the honey will absorb the heat. So it will protect their mucosal lining, the patient's mucosal lining, because the heat is transferred to the honey faster than to the mucosa. And after that, the patient can either swallow the honey or spit it out. So now I'm going to part 4, which is on the different honey products. And because I want to say that honey is not the only good things that the bee give to us, but there is also the royal jelly, the bee venom, the propolis, and the bee pollen. In this talk, I'm just going to cover on royal jelly, propolis, and bee venom. The wax has no activity. So now I'm going to talk about the bee venom. Bee venom for centuries has been used to combat baldness. So it's applied locally. It's very heaty substance, if I can use that word. So this is the local application that is used to treat hair loss done in mice. And you can see that 0.01% bee venom is useful for about 20 days. And it also prevents hair loss if it's combined with dexamethasone. Because the bee venom stimulates the expression of some growth factors that inhibit the expression of 5-alpha reductase, which converts testosterone to dihydrotestosterone. A very interesting constituent in bee venom is called apamine. It protects against Parkinson's disease and also Alzheimer's disease. I'm not going to go into it into detail because this research is reported to by others and not really my research. But I just want to share about the beauty of honey and other bee products. And bee venom, this is the latest research. It contains melatonin, and melatonin is useful against breast cancer. So what happened is it kills aggressive breast cancer cells and is not toxic to normal cells. Melanins will puncture hole in cancer cells and allow the penetration of adjuvant drugs like docetaxel to kill the cancer cells. So it acts together. is used as an adjuvant. This research is in the year of 2020. Okay, so then there is also the observation that bee farmers, they tend to live long, and we do not know why. So, and also telomere is associated with long life expectancy. So there is this question: do bee farmers, because they live long, do they have longer telomere? So that's the question. So interestingly, yes, we found that beekeepers have longer telomere. And in fact, for this research, continuous consumption of bee products for at least 1 year, it can be any bee product, increases telomere length by about 0.25 kilobase pair. So whether it's significant or not is seen among bee farmers who tend to live longer than non-bee farmers because of the constant stings or constant exposure to bee stings. So I'm not asking you to be stung by the bee, but like for these bee farmers, they get small doses, incremental doses every day over the many years, and it tends to help with the longevity. So then I'm going to also talk about propolis, which is a bee glue, which is the mixture that is used by the bee to create their hive, for which the honey is kept in for stingless bee, but for normal bee, the honey is kept in wax, which has no activity whatsoever. So propolis is richer in the stingless bee. Sorry, the honey taken from stingless bee has richer amount of antioxidant because it is stored in propolis, not in wax, and the antioxidant component of the propolis is being transferred into the honey as well. So there is the double dose there. So propolis has many medicinal benefits. It has caffeic acid phenyl ester, which can improve memory. And yeah. And it has anti-cancer effect, and it's also useful for multiple sclerosis. So again, propolis product also increases telomere length and is useful against aging. And this is one of our product, which I will talk about later. Oh yeah, here. So we want okay, so normally propolis, the bee farmers will throw propolis away because it cannot be processed. It's actually very sticky substance and difficult to process. So we found a method to process propolis for human consumption, and our product won the medal award for the International Technology Exhibition. This is done together with a company, with an industrial company. Okay, that's all I can talk about that part. And then I'll just touch on royal jelly. Royal jelly is the food for the queen bee only. Only the queen bee will consume the royal jelly. It is found to regenerate hippocampal granule cells, and it helps to increase collagen so that one looks young, and it has estrogenic properties. So it is used for premenopausal symptoms. So I remember my mother used to consume royal jelly when she was menopausal. So due to this reason, it's not useful for male because of the high estrogen properties that it has. So the last part of my sharing will be on Meliponiculture or stingless bee rearing. So this is me taking the trying to create a hive system at home. So normally when you go, when you meet the bees, it's good to wear white color because the bees, they prefer darker color shirt. So, uh, so you are encouraged to wear white to avoid the sting. If you don't like the sting, unless you are a bee farmer. So, stingless bee is just a little bit on stingless bee. Just now I was talking about the honey bee, the big size bee. Now I'm talking about stingless bee, which is the bee that you can rear at home. At home means along the corridor of your house or maybe in your garden. So it's a good pollination agent. In fact, when there is a fruit farm, the fruit production either double or triple when you have stingless bee around. In fact, Albert Einstein mentioned that if bees are extinct, human beings have only how many years to live? So the human population will be wiped out if the bee population is wiped out in 4 years. So that's how important the bees are. So I mentioned just now, stingless bee honey. Initially, we studied on tualang honey, then we converted to stingless bee because tualang honey is chopped down tualang tree is chopped down frequently, and also stingless bee has higher antioxidant properties, so we focus on stingless bee now. And why it has higher antioxidant properties? You can see that the honey sit on propolis pod, and the antioxidant properties of propolis is transferred to the honey in the pot. And then the value of the antioxidant increases. And you can see that the size of the honey bee is bigger than the stingless bee, where the size is about 15 millimeter compared to the stingless bee, which is only 3 millimeter. So this means that the stingless bee can go deep into the flower to get more nectar, to get a nectar-rich compound. But the honeybee, because it's big and bulky, it cannot go deep into the flower. So this is the system that we have at home, which you can also create at home. This is the log. It contains about 2,000 worker bee, which lives only for 20 days, and the queen bee, which live only for 7 years. Sorry, not only, which live for 7 years. It contains egg cell and also honey inside here. And whatever extra honey is kept in the box up here, and then that's where we take the honey. The box is called topping. And that is for aeration, for better ventilation, and the top part is to protect against the sun and the rain. So this is the system that we have. And you can see here, this is, I cannot interact with you. So I'm just going to say, this is a honey pot full of honey. And the one with the white spot on top is the matured honey. So once the honey is matured, the bee will cover the honey pot. And the one with the open hole is not yet matured. So don't take because it's going to be very watery and very sour as well. That is the egg cell. And this is propolis that we take for our research. So you can see here, so the biggest egg is the queen cell. So in one hive, there is only one queen. So this is the queen, there is the drone, and there is the worker bee. So the drones, they do not clean or build the honeycomb. They do not look for food. They do not help with the kids. They only eat and spend time mating the queen, and they mate in the air. And the drones are called the lazy wily or the flying sperm. So you can know that the lazy drone is actually the male, And the worker bee, the hardworking ones, are the female bee. I'm sorry about the males here, but this is the one that is occurring among the bees. So this is my daughter taking the honey. You can see that since it's stingless, it doesn't sting, but it can bite. Um, it's not that dangerous. Oh, so you can see my daughter sucking out the honey, and then she will put it in the bottle. Yeah, so we will get about 1 bottle per hive. So each colony may produce about 500 gram in a normal season. Normal season here means not rainy season. But in the flower season, it is a lot. Normally, we harvest once a month. And beside honey, we can also collect propolis for research. I know. So the bees will fly within 500-meter vicinity, and they will leave the hive around 7 a.m. and return around 7 p.m. But in between, there are many trips. So do not change they use GPS to come back to the hive. So do not change the outlet direction, because we try to change, and you can see that the bee, they will hit the wall of the lock, thinking that because they are going to the original outlet. Okay, so here you can see that the bees come back home. This is the outlet. These are my bees. So you can see they carry back two white spots on their back, and that is um, do you know what is that? Okay, I will show you what is that later. So these are the different mouth or outlet of the hive based on the species. The one that we have is the Itama species, and you can see that it makes a long outlet when there are predators around, like lizards. So they bring back the pollen for protein. The white spots just now are pollen. Then they also bring back the resin for their house, for their propolis house. And sometimes the bee farmers, they want faster product. So they try to help the bee by putting glass cups in the hive. So this is not really ideal, because the honey doesn't sit in the propolis pot, but instead sit in the glass cup. So the antioxidant amount is lower a bit. Because as I mentioned just now, the propolis pot also contributes to the antioxidant effect of the honey, because it leach into the honey. And the bees will bring back the nectar for their carbohydrate or energy source. So how does the bee communicate with the other friends? Oh, sorry, I use the word friends. Communicate with their colleagues to tell where the flower is. So they will dance. And then the angle of the dance will indicate the direction of where the food source is. And you can see here, so they are dancing. Now one person is dancing now, and the others are trying to measure the angle. And the faster the dance is, means the nearer is the food source. Okay. So what colored flowers do bee like? I'm not sure whether you can respond or not. Anyone want to shout? Is it the red one or the yellow one? Uh, okay, looks like we can't really interact now. Can we? Okay, it's the yellow one, because the yellow is red for the bees, because the bees' eyes are different from human eyes. Uh, And whose eyes are better? Better in the sense that the bees have bigger range. So we are seeing colors in our vision, which may be distorted as compared to the bees. So these are so if you want to rear bees at home, you need to plant the flowers that the bees like, and these are the ones that the bees like. Many actually, and it's not that difficult because some are found on the grass as well. So the bees visit about how many flowers per flight, right? It's about 100 flowers per flight. And we require about 4,000 flowers to produce 1 tablespoon of honey. So it's a lot of hard work. And a worker bee makes only 1/12 teaspoon of honey in her lifetime because the bees live only for 20 days. And when you eat 1 tablespoon of honey, you actually have benefited from 36 worker bees who have already lived and died. So sometimes the bees get tired, they fall asleep in the flower. So I'm finishing my presentation soon, it's reaching 1 hour. So just to cover on the challenges of honey research is the standardization of the compound. Like any natural product, we have issues with standardization because it varies from batch to batch across the geographical region. Like it depends on the water, the soil, the nectar, the quality of the flowers, and so on. And then allergic reaction is pretty common for bee product. So do test for allergy first. And the polyphenols are big molecules. They can't really penetrate the intestinal system well. So there is a poor bioavailability. And I mentioned just now briefly about tualang honey and the decreased number of bee population due to the rampant chopping down of tualang tree, sorry. So I would like to end by concluding by saying that among the honey that we investigated in Malaysia, sourwood and tualang honey, together with manuka honey, they confer good medicinal properties. And they have many have strong antioxidant properties, and we are still lacking in clinical trials. And stingless bee is something that warrant investigation because it has higher antioxidant properties. And I would like to promote beekeeping because it helps to ensure sustainability of the bees and can also give some financial income. And you can make sure that you consume pure honey, because it's from your own garden. That's all I have. Thank you very much. We can have some Q&A session now. Thank you. This is my email. Thank you. I will stop sharing.
Thank you so much, ma'am.
Welcome.
On behalf of the Society of Pharmaceutical Sciences and Research, we express our profound appreciation to Professor Gan Siew Hua for her outstanding presentation. Your scholarly insights, meticulous explanations, and rich research experience have significantly elevated the academic value of the webinar. We are deeply grateful for your gracious presence and esteemed contribution. Thank you so much, ma'am.
Welcome. Thank you.
Now, ma'am, there are some questions from the participants.
Okay.
The first one is, what are the major challenges in standardizing honey for medicinal use, especially considering the geographical and seasonal variations?
Yeah, so because they affect the nectar, the flower nectar, and look, we need to find what is the phenolic and flavonoid content that we want to standardize. So there are so many phenolic and flavonoid present in honey. So we need to identify a few fingerprints that should be available. And it's very difficult to standardize across the different countries because each country will have, or each geographical region will have their own unique phenolic and flavonoid content. Like I mentioned in my research, we found 9. So which one is best? But that's for Malaysia. What about other countries? It will be different. So that is the issue of standardization. I hope
Right,
you
ma'am.
will.
Thank you so much. Yes, ma'am. It's quite clear. The next one is, ma'am, how does adulteration impact the therapeutic efficacy of honey, and what
What, sorry?
analytical how adulteration impacts the therapeutic efficacy of honey,
How
and
what?
what
Sorry?
analytical method adulteration
How the identify oh yeah, that one is
adulteration? Yes,
the International Honey Commission recommend us to look for the pH, the 5-hydroxymethyl furfural content, the
right,
all the amino acid. So this is important because fake honey will not be able to contain the same constituent in the right amount. For example, the moisture must be within 20%, and then the proline, which is present in the bee saliva, must be present, and proline cannot be manufactured by human. So it
right, ma'am.
helps to separate from fake honey with the real honey, with pure
Right,
honey.
ma'am.
Yeah.
Right. Thank you so much, ma'am.
Yeah.
The next question is, ma'am, what recent clinical trials support the use of honey in managing chronic wounds, burns, or respiratory infections?
Respiratory infection, I didn't do any study on that, but burn, we can use uh, for burn, we can use honey locally, local application, but make sure that the honey has to be gamma irradiated first or sterilized because the honey may also contain some bacterial spores and things like that. So it has to be sterilized first before you apply locally on the burn area. Yeah. But I know
Right,
that for diabetes, like for example, honey, because it has high osmolarity, so it can pull out the water from the wound and it will allow wound healing to happen faster. So some people
right,
have
ma'am.
developed a transdermal patch of honey. Again,
Okay,
that has to be sterilized. Yeah.
right. Thank you so much, ma'am.
Okay.
The last question is, ma'am, what are the most promising future research directions in honey-based therapeutics, particularly for chronic diseases or immune disorders?
For chronic diseases, I think what is interesting is some people use bee venom to puncture at certain points, acupuncture points in their body, bee venom. For polio, for example. Uh,
Right.
so that one is interesting to be investigated. Yeah.
Thank you so much, ma'am. Thank you, Professor Gan Siew Hua, for your insightful responses and for addressing all the questions so comprehensively. Your expertise, clarity, and depth of knowledge have truly enriched today's session. We deeply appreciate the time and effort you dedicated to sharing your research, experience, and scientific perspectives with our participants. Your contributions have added immense value to the 102nd SPSR International Webinar, and we are honored to have had you with us today. Thank you so much, ma'am.
Thank you.
So, thank you, ma'am. So, here we come to the conclusion of this session. On behalf of the Society of Pharmaceutical Sciences and Research, we extend our deepest gratitude to Professor Gan Siew Hua for her outstanding presentation and the wealth of knowledge she shared with us today. Your session has truly enriched our understanding and inspired meaningful scientific discussions. We also thank all participants for your presence, enthusiasm, and thoughtful questions, which has added a great value to the program. The success of the 102nd SPSR International Webinar is the result of your continued support and engagement. We are deeply thankful to Dr. Shashi Alok, the National President of SPSR, for honoring us with his presence. We recognize and appreciate your commitment and guidance, and we offer our profound thanks for your significant input. We would also like to express our appreciation to our convener, Mrs. Monika Sabharwal, for her pivotal role in organizing this event. We extend our gratitude to all the SPSR team members for their collaborative efforts, which have broadened our understanding and fostered cooperative learning. Your collective contributions have made this webinar a success, and we look forward to the continued collaborations in advancing pharmaceutical sciences and research. With this, we formally bring the session to a close. We look forward to welcoming you to our future SPSR webinars and academic initiatives. Thank you and have a wonderful and productive day ahead. Thank you so much, ma'am. Thank you from Team SPSR.
We finish?
Right, ma'am, we
Sorry, I
can
think,
leave the session.
yeah, I think my line is not good.
Right, ma'am, we have finished the session. We may leave now. Thank you so much,
Okay,
ma'am.
thank you. Bye-bye. Thank you
Bye.
for running the session here, Dr. Pradeep.
Thank you, ma'am.
Yeah.